Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So today we will discuss about the ABO blood group system and RH group system. So the ABO blood group system is meaning of that some people have A blood group, some people have B, or some people have AB, or some people have O positive, O negative. So what that means and what is the basic biology behind that process. So here is the different person which contain a blood group A, B, and AB and O positive or O negative. So what that means? So here is the person A which contain a antigen A containing red blood cell. It means the antigen A are present is known as the A blood group. So we are giving the name on the basis of antigen. So here is the B. B is the contain B antigen, and while the AB is contain both antigen A and B antigen, which that is antigen is bind with the antibodies in our blood to give a complex. So we will not discuss about that, but just understand the ABO blood group system. So here is the O positive and O negative, which that is also contain a H antigen, which that is also present in the AB group, but RH antigen is present in O positive while RH antigen are not present in O negative. So that is why it is known as the O negative. So let's begin to understand from red blood cell is anucleated when it means it will not produce the antigen during the anucleated. So how it's produced? So let's begin to understand. Here is the erythropoietic precursor cell stem cell. Stem cell will convert into the erythroblast after the proliferation of the erythropoietic stem cell, and erythropoietic stem cell convert into the erythroblast into the normoblast. Normoblast contain a nucleus well. So here is I am drawing the structure of the normoblast, which that is produce or represent the antigen in the cell membrane of the precursor of a red blood cell, which that is not mature, it is immature in the bone marrow. So here is the 90 chromosome in our nucleus in throughout our body. For example, the normoblast will be activate the antigen gene are present in the region of 90 chromosome. I will not mention the origin of the gene, but you can understand, and that's transcribe, and this transcription to become a messenger RNA after the basically transcription. So the transcription to produce the messenger RNA fucose transferase enzyme, which that is the part of protein. After the translation of messenger RNA produce the fucose transferase enzyme, and fucose is the glucose. So remember about that. So that is the autosome. So it means fucose will bind with the fucose transferase enzyme to give the oligosaccharide one by one on the cell membrane of the normoblast. So after this process, so the RBC will be proliferate and anucleated become. After the nucleus will be dysfunction and nucleus will be dissolve, so you can say that, and red blood cell will be produced and contain a oligosaccharide. So what that means? So that is the overview. Let's begin to understand the blood group A. So here is the blood group A, which contain a basically gene on the chromosome female and male containing a pair of chromatin are present in the and same centromere, but I am drawing the separately to understand well. So here is the chromosome 9. So this chromosome 9 contain a locus, and locus contain a gene, which that is both genes are allele to each other. Allele to each other mean the companion to each other, which that is one is dominant and one is recessive, but their character is same. So the messenger RNA will be produced, and this messenger RNA will produce in the A blood group system to produce N-acetyl glucosamine transferase enzyme production by the messenger RNA into the translation. So the N-acetyl glucosamine will be transferred on the upper oligosaccharide to bind with the NAG or NAGA, you can say. So this NAGA will be attached on the upper side of the oligosaccharide, which that is come from the 90 chromosome, and that is bind, and it means it is we are giving the name is blood group A. So that means it is which that it can be react with the antibody against this antigen type. So that means it will be clotting occur during the IgM IgM antibody will be bind. So that is the another topic we will not understand. So here is the blood group AB, which that blood group B which produce the chromosome 9, same mechanism, the female and male homologous chromosome which that contain a gene locus, a variant gene, for example A or B. So O and B, sorry. So the B will be dominant. It means the messenger RNA will produce and B antigen are present. That is the galactose transferase enzyme used. So the galactose will pick from the cell cytoplasm and give to the H substance, or you can say the H substance which that's come from the chromosome 19, which that is bind with the galactose, and so this galactose binding is known as the B antigen, which that is the basically B antigen. This antibody bind with the B antibody. So that is the structure of that of the B blood group B. While here we will discuss about just overview of the chromosomes. So the chromosome is contain a DNA and histone protein. So the DNA is the genetically molecule, macromolecule, which that is expressed with containing a gene. So here is the recessive gene is one chromosome in the alleles of one allele is dominant and one allele is recessive. So the recessive allele will not produce any substance, while the dominant allele can produce substance A, for example. So that means that is the chromosome. Half is come from the parent, half is come from the female, and half is come from the male, which that is combined together a 46 chromosome throughout our body in every cell, each cell. So here's the blood group AB. Let's begin to understand what that is meaning. So the B is come from the B gene. B containing gene chromosomes come from the female, while the A antigen containing chromosomes come from the father. So that means female. So that is combined together to give a AB combination of that alleles. And also chromosome 19 will also produce the H antigen, and that is galactose NAGA transferase enzyme is also used for the, and galactose is also, and it means the N-acetyl glucosamine and galactose is combined together to give a two type of antigen. So that is why it is known as the A and B antigen, AB blood group. So that means the here is the galactose also bind with the H substance, while the H substance also bind with the glucosamine due to the translation of the NAGA T, mean the N-acetyl glucosamine transferase enzyme. So let's begin to about the genotypes and phenotypes. What that means. So the genotype is the separate discussion, while the phenotype is the separate. So here is I am drawing the chromosome, which that is one is come from the female and one is the male. So for example, the allele to each other. So that means the A antigen containing chromosome and O antigen containing chromosome. So it means the O will be recessive while A will be dominant, but their gene is the AO and AA, or we can say. So that is the heterozygous or homozygous condition. Hetero means the AO, while the homozygous mean the double A, which that both allele are equal to each other, while the BO is the same another. So the OO is the homozygous, BB is the homozygous, but AB is the heterozygous, but it is co-dominant between complete a codominant, basically both chromosome will bind. So, for example, we will discuss about the blood group O. What that means? It means the non-functional chromosome A, B are present. Female is come from the A home locus, while the B containing B antigen, which that is both are inactive, genetically non-functional. So that means epigenetic mutation can be occur. So it means that both alleles are recessive or both allele is dysfunction. So that what that means, the 90 chromosome which that's come from the female and male, which that is both are allele to each other, produce H substance. So that H substance is the functional transferase enzyme used to basically functional transferase enzyme not and produce due to the A and B gene of allele are no product, giving the no product. So the H substance is already present, still present. So H substance containing O blood group is known as the basically O blood group. So let's begin to the RH group system. We will not forget about that. So the RH group system means it is the RH positive antigen, RH containing gene which that is activate to produce the protein. That is protein is known as the RH protein, which that is our view. Next video we will discuss about the detail. So it means the A positive, AB positive, O positive. That is basically O positive is the universal recipient. Why? Because it contains all type of basically antigen, which that is basically it can be bound with the A positive, AB positive. So that means if positive, so that means A negative is not. It can be allowed to A negative, AB negative, but on the other hand, the RH antigen negative, which that's contains universal donor, it can donate because it is not antigen any antigen present in the surface of the red blood cell. So it can donate because it is not produce the reaction against the antigen to produce antibodies. So that's why it is the donor, while the recipient O positive due to the it's contain a antigen, and that protein is basically that protein is if we'll give to another O negative. So the O negative, if we give the antigen containing O positive, so it means the agglutination will be occur, and the all red blood cell will be damaged. So that's why RH group is important for us. So thanks for watching. Please make sure to subscribe and give the suggestion for a new video, and I am waiting. So bye.